So, the principles of antimicrobial therapy. The introduction of modern drugs to control infections was a medical revolution in the 1930s. The 1930s, guys. Antimicrobial drugs have reduced the incidence of certain infections, but they have not eradicated infectious disease and probably never really will. Today, doctors are worried that we are dangerously close to a post antibiotic era where the drugs we have will no longer be effective. So, the goal of antimicrobial chemotherapy, when we think of chemotherapy, we typically think about, medication you give to patients with cancer. That's not technically what chemotherapy is. Chemotherapy is a broad term that just means any medicinal medication. So this is the goal is to administer a drug to an infected person that destroys the infective agent without harming the host cells. So this drug has to be able to get um, into the body and reach to where it needs to get to. It has to be easy to administer. It has to be absolutely toxic to the infectious agent, but not to the actual host itself. It needs to remain in the body as long as needed, and then be safely and easily broken down and then excreted from the body. So this is the characteristics of an ideal antimicrobial drug. You can read it here. It goes into more detail than we said before. One of them being reasonably priced would be ideal. Some medications are super expensive, but we don't want it to be. So the best medications are the ones that are safe, that work well, that don't have a lot of side effects, that also don't cost a lot. Those are the best medications, and we give those out like gangbusters. So the terminology of chemotherapy. So this is a little helpful as far as with the term chemotherapy being in the name. So a chemotherapeutic drug is any chemical used in the treatment, relief, or prophylaxis of a disease. Prophylaxis is the use of a drug to prevent imminent infection of a person at risk. So this is something um, or a situation where a person doesn't have the disease, but they're at great risk for the disease. So, the patient has risk factors for this particular disease. They're of this age, they're this weight, they're this gender. So it's risk factors, but it's not actually, them having the disease. Antimicrobial chemotherapy. So this is the use of chemotherapeutic drugs to control infection. So that's a specific type of chemotherapy. Antimicrobials themselves are all inclusive term for any antimicrobial drug, regardless of its origin. So this means antimicrobial could be antifungal, antiprotozoal, antiviral, antibiotic, etc., etc. So antibiotics are substances produced by the natural metabolic processes of some microorganisms that can inhibit or destroy other microorganisms. Semi-synthetic drugs versus synthetic drugs. So semi-synthetic are drugs that are chemically modified in the lab after being isolated from a natural source. So we actually get it from a natural source, and then we kind of tweak it, usually in order to make sure that we get the effective dose to the patient without giving them too much. That's one of the problems with holistic medicine. It's great because, to be honest, some of the medications you're getting are coming from the medicines that you're getting from holistic medicine. It's just the difference is you could easily overdose or underdose if you're taking it from nature, because you really honestly can't be sure how much to use or how much is in that particular leaf, for example. A synthetic drug is, on the converse, is drugs produced entirely by chemical reactions in the lab. So narrow spectrum versus broad spectrum is exactly what it says. Narrow spectrum is also called limited spectrum. So this is antimicrobials effective against a very limited array of microbial types. So it may only be effective, for example, against only gram-positive bacteria. So gram-negative wouldn't be affected by a narrow spectrum gram-positive antibiotic. Versus broad spectrum is an extended spectrum. It can go against a big, huge variety of um, different microbials. So gram negative, gram positives. It could be some other um, um, what is it? Not the spore forming. What is it called? The heat resistant, like Mycobacterium tuberculosis. So it can. It's just a broad spectrum. It can go against a lot of different things. So the origins of antimicrobial drugs. Antibiotics are common metabolic products of aerobic bacteria and fungi. So they're produced to inhibit the growth of competing microbes in the same habitat. They're derived from bacteria in genera Streptomyces and Bacillus. and molds in the genera Penicillium and Cephalosporum. So, identifying the agent and starting treatment. So, before the actual antimicrobial therapy can begin, three factors must be known: the nature of the microorganism causing the infection, so we got to know what it is, the degree of the microorganism's susceptibility or its sensitivity to various drugs. So, this goes into when they do cultures and sensitivities of the different, you know, particular areas that we cultured, so like sputum sample or a wound sample or a blood sample, and we actually look for that actual microorganism, and then we'll do some testing to see how sensitive it is to certain medications that we have available. And then the overall medical condition of the patient. So again, if the patient's young and healthy with no other comorbidities, the patient, we can give them, I won't say a simpler treatment, but an easier treatment, send them home, we're good to go. Versus if they're older and have all these other health problems, they may need to be admitted to the hospital and get the same medication, but get an IV because it gets more direct into the blood and it works faster. So, identification of infectious agents should begin as soon as possible. So, it should occur before the antimicrobial drugs are given, before their numbers are reduced. So, the best nurses are the ones that are anticipating the physician's moves. They go in, the patient's coming in, they're complaining of, burning when they urinate and frequently urinating. So, that nurse, hey, they should be telling that patient, you need to pee in this cup before the doctor comes in and chucks them full of antibiotics. That way, we can send it off to a lab, see what's going on in that urine sample, because it's fresh. It hasn't been tainted by antibiotics that are now trying to kill those microbes, and does a good job very quickly and does it well. But then we can actually go, and even let's say the patient gets admitted, because let's say they're elderly and that's why we admitted them with UTI, we're going to treat them for 7 days. But what we can do is we can switch their antibiotic treatment, because, hey, maybe what we're giving them is a broad spectrum, by the way. So we'll give them something broad to cover most things, but then we can switch it after, 2 days, the culture comes back and it's like, oh, it's E. coli. We can give a more specific medication that kills E. coli, so a more narrow spectrum. We don't have to. We can keep them on the broad spectrum, but we use a lot of broad spectrums, so we try not to use it more than we have to. And it's better to use a narrow spectrum that's going to kill specifically what's there. So that's one thing we could do. Or another reason is because we can find the medication where we have them on shows that it's not susceptible to the medication they're on. So we have to switch them to something that will actually treat the infection. Okay, so now let's get past that point. So, direct examination of samples is a rapid method for detection. Doctors often begin therapy on the basis of just immediate findings and informed guesses. So, we don't delay treatment because we're humane. We just give medication prior to basically knowing for sure what something is or what the causative agent is, because we don't want to just torture somebody and say, okay, sorry, you're going to have to live with this infection and live with this pain for the next 3 days until we get a definitive source. We're not going to do that. And epidemiological statistics also may be required. So, testing for drug susceptibility. Testing is necessary for the following organisms: Staphylococcus species, Neisseria gonorrhea, Strep fascialis, however you say that, faecalis, and aerobic gram-negative intestinal bacilli. So these are going to be things like Shigella, Salmonella, Escherichia coli, things like that. Testing for fungal or protozoal infections is difficult and unnecessary. Testing is unnecessary for organisms known to be susceptible to penicillin G unless the patient is allergic to that medication. So, Kirby Bauer technique. I'm sure you probably heard of Kirby Bauer. Maybe it's just me because I've been in school for forever. I've heard of it many times. But the Kirby Bauer technique. So, this is a surface of an agar plate. It's spread with bacteria. So, we spread it all across. And then we put these small discs containing a prepared amount of antibiotic on that are placed on the plate in different areas, not too close to each other. So what's going to happen is you have a bacteria that's on a plate. So this is probably from, like I said, some sputum sample. We've taken that sputum sample, as we said in previous PowerPoint slides that we've gone through. You take it, and you're going to streak it in a plate in those 4 different ways to grow colonies to isolate the bacteria. You're going to isolate the bacteria, and then you're going to take it and put it on this plate. So it's pure bacteria from whatever culture we got it from. And then you're going to put the antibiotic, they're like little tiny paper discs, and you put it on this plate, okay? And then what happens is the antibiotic, if it works on that particular bacteria or microbe, is going to form what's called a zone of inhibition. So it's actually going to kill the actual bacteria that is in the vicinity of that disc. So you're going to have areas of clearing around the disc. And the greater the area of clearing, the more susceptible that bacteria is to that particular antibiotic. And so we do this, and again, it's that zone of inhibition that's surrounding the disc. We're going to measure it. So actually measuring it will tell us how susceptible it actually is. So an antibiogram provides data for drug selection, and we'll see that in the next slide. And this method is less effective for anaerobic fastidious or slow-growing bacteria. So this is the results of a Kirby-Bauer test. So it's showing the different drugs on the left column, and then it's showing you the zone sizes and millimeters required. So again, this is we're measuring in millimeters. So actually, you'll see on the next slide how they're measuring the actual zones of inhibition. You're actually measuring from the entire diameter around the particular paper disc, okay? So susceptibility versus resistance, and so it's showing you in millimeters what it will be. So if it's greater than 13 millimeters, then we know it's susceptible to bacitracin, and we can use bacitracin to treat. If it's under 8 millimeters, then we know it's resistant to bacitracin, and we don't want to use it. If it's in the middle, it's kind of ambiguous, and so it's called this intermediate area. So you can arrange. So if you look at erythromycin, the actual result for Staph aureus on this test for erythromycin was a zone of 15. Well, above 18 is what we would need for it being susceptible to use it. And below 13 would be we can't use it at all. So it's that I, which if you look at the bottom, it tells you what the letters stand for. So it's intermediate. So it's one of those ones, I'm gonna be honest, we probably don't want to use because is it going to be effective? Maybe, but how effective? Probably not too great. So obviously on this particular evaluation, we have 1, 2, 3, 4, 5, 6 different medications that are actually susceptible to basically having that zone of clearance, which means that those medications will work on Staph aureus. So we could use those and know that they're going to work, okay? And remember, too, the thing about it is, it's not only the particular bug. So Staph aureus is a bacteria. So it's not just that bacteria. It is looking at, whatever particular strain of this bacteria or particular microbe. So I could have E. coli and you could have E. coli in a UTI. They can do the culture and sensitivity, and then they can have this test, and our Kirby-Bauer test results are not going to be exactly the same. So it's specific to the person. Otherwise, all we would have to do is say, oh, they have E. coli. We give this medication. We're done. Well, actually, they have to look at what strain of E. coli you have and then figure out what strain or what medications are going to work for the E. coli you have. So if you look at this, it's showing the zone of clearance. So everything in that top left box is saying the zone of inhibition. So that's that big yellow area. You have the antibiotic carrier, so the disc itself. So each one has a different letters and numbers, and it's telling you the actual medication plus the grams. Also, some of these medications we don't actually use, but they have we use them in the laboratory in this sense because it's easy, and then we actually usually give you the medication that falls into the same class that we more commonly use. So anyways, so we have that. So if you notice, what is this? Cefotaxime all the way on the left has a pretty big zone of clearance here. I probably shouldn't use that one because I don't think it tells us exactly what the number is. So let's look at the top where they're actually measuring it. So it's enrofloxacin. I have no idea what that is. So it has a zone of clearance of almost 30 millimeters. So you see here they're measuring at the center diameter, and so it says anything over 22 millimeters is sensitive, excuse me. So in this case, it's going to be sensitive. So it's showing you the different rings, and so they're actually showing you whether it's resistant, sensitive, or intermediate. If you see over there in that picture on the right, so those two zones of inhibition actually crossed over into each other. So we can actually still measure it. You just measure it on the side that's not touching. So more like vertical in these particular situations to kind of figure out what the actual zone of clearance is on those particular antibiotic discs. So we also have dilution tests for tubes. So this is more sensitive and quantitative. So we actually get a quantifiable number. And so you're going to get this with tube dilution test over the Kirby-Bauer test. So the antimicrobial is diluted serial in a series of tubes, and we'll see that in the next slide. So then each tube is inoculated with a small uniform sample of the pure culture. And then you're going to have what's called a minimum inhibitory concentration, or the MIC or MIC. This is the smallest concentration, which is the highest dilution, of a drug that visibly inhibits growth. Okay, so it's useful in determining the effective dosage and providing a comparative index against other antimicrobials. So what this is showing, if you look at the top, you actually have a control. So it's a negative control. So basically, there's no antibiotic in it. And you make sure that we like to have controls because it lets us know that things are working. So if you have bacterial growth in the negative control, then we know that bacteria is there. If nothing grows in the negative control, then that's a problem because then it's like your original sample didn't have bacteria. So then we could throw the whole test out because it didn't work. Or something was wrong with that first culture for some reason. So anyways, as you see the other test tubes, it's showing you have 0.2 micrograms per milliliter of the actual medication, and then the actual amount of medication goes up. So obviously, if we have more of the medication, we expect it to kill that particular microbe. If you have less, it depends on, how little of it can you use. So remember, we don't want things to be harmful to the person or to the host. So we want to use something that's effective, that's going to kill off the particular microbe, but we want to use as little as we possibly can to still be effective. So it's showing you the little dots is growth and no growth is the yellow. So the minimum inhibitory concentration is going to be the one that has no growth. That is the lowest dose, okay? From here, it kind of looks like the 6.4 tube doesn't have any growth. It seems like there's a little bit in 3.2. So the minimum inhibitory concentration would be 6.4. So if you look at the bottom, it's showing you the negative control at the top, okay? And then it's showing you different particular these are actually antifungals. So fluconazole, itraconazole, ketoconazole, voriconazole, capsifungin, all these different ones. Amphotericin B, that's the one under the negative control. I think 5-fluorocytosine or something. I forget the 5-FC. But anyway, so it's showing you the increasing concentration as you go up. So going from the reddish to the more purple blue side, it's showing you where there's finally no growth. So it's a lower dosage on the reddish side and a higher dosage on the blue side. So for amphotericin B on well 5 or tube 5, that's the minimum inhibitory concentration. So it's actually color coded. It's showing you that when it's completely blue or that purple, there's no growth. So they actually have a pH indicator showing you in this particular sample. So for fluconazole, it's well 12 where there's no growth. So whatever concentration that is going to be the minimum inhibitory concentration. Voriconazole is number 4. So anyways, it's showing you the first tube's concentration and then going up from there. So this is the tube dilution test. So it's giving you a quantifiable amount of medication at its lowest inhibitory dose that you can use to get rid of the infection. So, the in vitro activity of a drug is not always correlated with the in vivo effect. So, in vitro is like in a test tube versus in vivo is like in the live person. So, failure of antimicrobial treatment is due to the inability of the drug to diffuse into that body compartment where we need it, resistant microbes in the infection that didn't make it into the sample collected for testing. So remember, sometimes you have a resistant form of the microbe, like a spore, along with the actual particular vegetative cell. And an infection caused by more than one pathogen, so a mixed infection, some of which drugs are resistant to. So you may have two particular pathogens or three in a particular wound site, and you want to treat it, and the drug only affects two of the three, so you're not going to get complete healing and infection resolution based on that one antimicrobial medication. So the therapeutic index is the ratio of the dose of the drug that is toxic to humans as compared to its minimum effective dose. So basically, we don't want this to be a ratio that's close to each other, because if it's like that 1.1, that means that the numerator, so the effective dose, let's say it's 10 milligrams, and then the the toxic dose, let's say is 10 milligrams, and the minimum effective dose is like 9 milligrams. That's a super close window. So we really don't want to play that Russian roulette game. So those medications we're not using as frequently, versus something with the therapeutic index of 10, which means, let's say it's 100 milligrams for it to be toxic, but only 10 milligrams to be effective. Well, we're not even giving you any, we're close to that 100 milligrams. So it's a safe medication. We can give it. We're happy. The drug with the highest therapeutic index has the widest margin of safety. So the physician must take a careful history before prescribing an antibiotic. This is really important. Histories are huge. Pre-existing conditions that might influence the activity of the drug need to be known. So we don't want to, we want to basically give the best medication we can without causing other issues. History of allergy to a certain class of drug is super important. I'm allergic to ACE inhibitors. If they need to give me, if I have some condition that comes up, the ACE inhibitors are great to treat. There's probably some other medications they can give, but they need to know, let's not cause this death situation in this patient. Let's give her something else. So underlying liver or kidney disease. So remember, these patients cannot, if they have a poor liver, they can't, they don't go through that first pass well. So their medication is at a higher dose than what we want it to be at. And then kidney disease, the medication doesn't get excreted probably in the way it should. So it actually circulates more in the body. So it's like getting a higher dose. So you have to be careful, and you have to dose for patients with those problems. Infants, the elderly, and pregnant women require special precautions. So we don't want to hurt these particular groups of individuals. Intake of other drugs can result in increased toxicity or failure of one or more drugs. So some medications you have to be careful with, certain, not just other medications, but food and drink. So grapefruit is one I remember from med school. There's certain medications you have to be very careful with. Genetic or metabolic abnormalities and the site of infection, the route of administration, and the cost all matter as well. Some patients can't afford it. They're going to say they'll take it, but they can't. The route of administration, if it's eye drop or oral medication, it's a little easier for the patient to take it home. If it's not that, the patient's probably not going to want to take a shot if they don't have to. So are they going to be compliant? And if it costs a lot, they definitely aren't going to be compliant. But they'll lie and tell you they will. So, two hypothetical cases. You have an elderly alcoholic patient with pneumonia caused by Klebsiella, which is a bacteria, and complicated by diminished liver and kidney function, requiring a parenteral drug administration and a history of allergies to penicillin. And then you have a cancer patient with severe systemic candida infection. Even when all of the information is in, the final choice of a drug is not always easy or straightforward. So, the goal of antimicrobial drugs is to disrupt cell processes or structures of bacteria, fungi, and protozoa, to inhibit virus replication, interfere with the functions of enzymes required to synthesize or assemble macromolecules, so the carbs, proteins, nucleic acids, and fats, destroy structures already formed in cells. Selectively toxic means to kill or inhibit the action or synthesis of molecules and microorganisms, but not in vertebrate cells. So, drugs with excellent selective toxicity block the synthesis of the bacterial cell wall, and these are drugs in the class of penicillins. Humans lack the chemical peptidoglycan, and so we're unaffected by drugs like penicillins that are going to selectively be toxic to that wall. Most or drugs most toxic to humans are drugs that are going to act on similar structures that are common to our host cells and to infective agents like the cell membrane, because our cells do have membranes. As characteristics of the infectious agent are more and more similar to the host cell, selective toxicity really honestly becomes a lot more difficult to achieve. So, goals of chemotherapy: identifying structural and metabolic needs of a living cell and removing, disrupting, or interfering with these requirements. So, antimicrobial drug categories: you're going to have inhibition of that cell wall synthesis. So, we've already talked about that. Inhibition of nucleic acid structure and function. Remember, the nucleic acid that's actually inside of the bacteria or the microbe itself. Inhibition of protein synthesis, so the actual proteins that these bacteria can make. Interference with the cell membrane structure and function, and inhibition of folic acid synthesis. So this slide is just kind of going back over some of the areas of the structure of the bacterial cell and where some of these medications work. So this is literally how we come up with antibiotics. So you have classes, and the classes are going to work in the same similar way. They all have different specifications amongst the different drugs in the class, but they all kind of have the same overall mechanism of action. So it's showing you particular cell wall inhibitors, particular medications that are going to affect the cytoplasmic membrane, ones that are going to affect DNA or RNA, so the actual nucleic acids themselves. So it can either inhibit replication and transcription, or it can inhibit the RNA polymerase. You have folic acid synthesis, which is something else that the cells can use. And then you have protein synthesis inhibitors that act on the ribozymes. So it breaks it down into actually what subunit. So is it the large subunit, the small subunit, or is it both? So I skipped over the previous slides. Make sure you go back over them. They were just going in a little more detail of a few more medications that fell into the group that we talked about on the slide with actual bacteria. So broad spectrum and narrow spectrum drugs I've already talked about, and the differences between the two. Penicillins. So the original penicillin was a narrow spectrum and susceptible to microbial counterattacks. The molecule has been altered and improved upon over the years. So later penicillins overcome the limitations of the original molecule. So here's a picture showing spectrum of activity for antibiotics. So it's showing as it goes kind of across the board. So mycobacteria, remember, acid fast, and then you have gram negative, gram positives, chlamydia, which is a different kind of intracellular particular bacteria, and then rickettsia. So basically, it's showing that it's looking at different groups of medication and kind of how far they spread across. So if you notice tetracyclines, which was the example they used on the previous slide for broad spectrum, it covers rickettsia, chlamydia, gram positive, and gram negatives. It doesn't cover mycobacteria, which is kind of its own little thing. So you see that isoniazid, on the other hand, is very narrow spectrum because it only covers tuberculosis. So anyway, so they're just showing you different spectrums of the medication on this slide. So this one's actually showing characteristics of the penicillin drug. So it's actually a whole group. So you have penicillin G, which is the original. So it is narrow spectrum. If you notice, it kind of goes down narrow, 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 and then broad, broad, and very broad. So these are different medications that fall into this penicillin-like group. So penicillin G, penicillin V, which are given through different means. Methicillin, nafcillin, ampicillin. Ampicillin is a very common medication they give to kids and to women who are pregnant with group B positive strep on their cervix. Um, Amoxicillin, so that's a common medication. I'm sure many of you have probably taken it. So that's broad spectrum as well as ampicillin. And then this third class, which we don't use very much, is also very broad as well. So they're just kind of showing you the medications in this class, whether it's narrow or broad. And how they vary. So if you notice, the beta lactam ring is the same on all of them, and then it's that extra functional group that's hanging off that makes it a little different. So antibiotics and biofilms. So bacteria and biofilms behave differently than when they are free living. So it's often unaffected by antimicrobials. Um, Antibiotics often cannot penetrate the sticky extracellular materials surrounding biofilms. So bacteria and biofilms express a different phenotype and have different antibiotic susceptibility profiles than the actual free bacteria that is actually congregated and causing this biofilm to occur. So, biofilm treatment strategies, interrupting the quorum sensing pathways. So, you have things like daptomycin has shown success. It's a very powerful antibiotic. Adding DNase to antibiotics aids penetration through extracellular debris. So, this is an enzyme that's going to break down the DNA. Impregnating devices with antibiotics prior to implantation. So, this is a prophylactic way of treating possible biofilms. So, when we're going to insert, something like a stent into a coronary artery, you could actually insert, and often they will insert drug-eluting stents into the coronaries and actually other parts of the body. And so it slowly, over a matter of a long, long, long period of time, will emit antibiotic drugs in order to stop the production of biofilm in the first place. So some antibiotics cause biofilms to form at a higher rate than they normally would. So we still have to kind of be careful with treatment. So agents to treat fungal infections. So eukaryotic cells that represent special problems in chemotherapy. So drugs designed to act on bacteria are ineffective against fungi because it's not the same, right? Not the same cell structure, so it's not going to affect the same way. So similarities between fungal and human cells means that drugs toxic to fungi will harm human tissue. So that's one of those unfortunate parts, again, where we don't want it to be similar, but unfortunately, fungi in ways are similar to our cells. So, the class of antifungals isn't as large as antibiotics. And some of them are really strong, but can be very damaging to the patient and have side effects. So only a few agents with special antifungal properties have been developed. So here's showing agents to treat fungal infections. So amphotericin B is the very, very strong one. We have the azoles, which you're probably very common with the azoles. So this is fluconazole, ketoconazole, miconazole, and clomitrazole. So they're actually fluconazole is the antifungal that's used for vaginal yeast infections. So fluconazole is actually diflucan. So that's the oral medication, but it's also, this group of azoles is also the cream based medications that they use for the topical treatment for yeast infection as well. I'm sure the guys in the class are just loving that part, right? You have also mycofungin and capsulfungin. So these are other particular antifungals, and then flucytosine. So anyways, they just all have different mechanisms of action. If you just want to read there about what those are, just be familiar that these are antifungal medications. So protozoal infections. So they're going to have obviously a different group of medications to treat against protozoal infections than fungal infections and bacterial infections. So quinine is the principal treatment for malaria for hundreds of years. So since then, it's been replaced by quinolones like chloroquine and primaquine. So several species of Plasmodium, which are protozoals, and many stages in its life cycle means that no drug is universally effective. So remember that protozoals have, different stages that they go through. Their cyst form, their trophozoite form. And so when it's in one form, a medication may be able to fight against it, but not while it's in the other. So metronidazole, this is a very, very common medication. Um, I remember the mnemonic in med school was get gap on the metro, and then we added in rosacea. So get gap on the metro meant that it was get, so Giardia lambia, the E was Entamoeba histolytica, and the T was Trichomonas vaginalis. I forget what gap is at this point. I guess I could look it up, but y'all probably don't care. But get gap, and then so gap was for three other particular microbes, and then on the metro meant using metronidazole for it. And so rosacea actually can be treated with metronidazole as well. So it's a widely used amoebicide. So it's going to treat intestinal and hepatic diseases. So I've already named some of those. And then other anti-protozoal activities or medications, excuse me, with anti-protozoal activities are these down here. So out of these, tetracyclines are very common, and sulfonamides are also very common. So agents to treat helminthic infections. So remember, these are our worms. So the challenges of anti-helminthic, I don't know, just you know what I'm trying to say, drug therapy. So you have flukes, tapeworms, and roundworms that are larger parasites, and their physiology is much more similar to humans. So again, now we have that problem of wanting to affect the worms but not affect us and our physiology. So blocking reproduction does not affect adult worms. So it affects the other stages, but not the adults. So the most effective drugs immobilize, disintegrate, or inhibit the metabolism of all stages of the life cycle. So mebendazole and albendazole inhibit microtubules of worms, eggs, and larvae. Please don't get these confused with the azoles that are antifungal, but they happen to have azoles at the end as well. So we have pyrantel paralyzes the muscles of intestinal roundworms. You have praziquantel and ivermectin. Out of these, the bendazoles, and actually that is like how you would characterize it. It's not azoles, they're bendazoles. So anyway, so those and ivermectin are most commonly used to treat helminthic infections. So, agents to treat viral infections. So, the unique challenges with viruses. So, an infectious agent relies on the host cell for the vast majority of its metabolic functions. So, disrupting the viral metabolism requires disruption of our cellular metabolism. So, measles, mumps, and hepatitis are prevented through the use of vaccines. So, that's one way that we've addressed the challenges. A lot of viral medications we actually have vaccines for. So we're preventing them from the patients from getting the infection in the first place, so we don't have to have antivirals. I will say, out of all of these medications, the antivirals are I don't want to say the crappiest, but we haven't really worked the best with those. We have some, and they do stuff, but they're not the best. An example is Tamiflu. So that's an antiviral medication, and I mean it works sometimes, but a lot of times it doesn't do much. So AIDS, influenza, and the common cold attest to the need for more effective medications for the treatment of viral infections. So we get these, at least the influenza and the cold, all the time every year, and we just don't really have anything to treat it except symptomatic. So the major modes of action of antiviral agents is barring penetration of the viruses from getting into the host cell, blocking transcription and translation of the viral molecules, preventing maturation of the viral particles. So DRACO, or double-stranded RNA-activated capsase oligomerizer, is a breakthrough antiviral treatment currently being tested. So these are some antiviral medications, if you notice here, and they all act differently. So again, this has to do with knowing and understanding the microbe itself, understanding how it infects the cell, how it gets in, what it does when it gets in the cell, and those are the targets that we're going to use, obviously, for creating these medications. So that's why microbiology is so important. So with viruses, we can inhibit their entry. We can inhibit the nucleic acid synthesis that's going to go on inside our cells, and we can inhibit actually it leaving the cell. Okay, so it gives you the medications that fall into each group. So let's see, what do we have that? Oseltamivir. So this is Tamiflu, right? So we use this to block the entry of influenza virus from getting into our cells. We have acyclovir, and acyclovir is a common medication for herpes. We have ribavirin, zivivudine, lamivudine. All of these are HIV medications. Indinavir and sequinavir. All the navirs are usually medications that are used for HIV patients. So we have a lot of them. We've actually come a long, long, long way with the HIV medication. So HIV patients live really, really long, long lives now, really long, healthy lives because we've come so far with um, anti HIV medications. In the past, the side effects used to be so bad that people, really didn't even want to take the medication in the first place because it was like, why, if I'm going to be in a hospital bed, because the side effects suck so bad. But now we've actually cut down on some of those side effects a lot, cut down on the cost. I mean, the cost is still high, but it used to be crazy, crazy higher than it is now. So we've, like I said, we just come a long way, and we have a long way to go with antiviral medications. Drug resistance. It's an adaptive response in which microorganisms begin to tolerate an amount of a drug that would normally be inhibitory due to the genetic versatility and adaptability of microbial populations and can be intrinsic as well as acquired. Resistance to penicillin developed in some bacteria as early as 1940. Mind you, penicillin was discovered in 1928. So that's not that long. That's 12 years later. In the 1980s and 1990s, scientists began to observe treatment failures on a large scale. Microbes become newly resistant to a drug after one of the following occurs: spontaneous mutations in critical chromosomal genes, acquisition of entire new gene or sets of genes via horizontal transfer from species. So, we've talked about both of these in previous lectures and how this phenomenon can occur. So the development of drug resistance, chromosomal drug resistance. So usually results from spontaneous random mutations that will happen in that genome. Slight changes in drug sensitivity can be overcome with larger doses of drugs. This isn't always the case though. And then you have things called persisters, which are slowing or stopping of metabolism so that the microbe can't be harmed by the antibiotic. So then now we have resistance through horizontal transfer. So this slide and the previous slide were the two examples that they mentioned previously as to how we get resistance. So you get those resistance or R factors, and these are plasmids that contain the antibiotic resistant genes. So remember, one's going to develop it, and then over time, it's going to transfer it to various bacteria through time. So this can be transferred through conjugation, transformation, or transduction. If you remember those three and how conjugation is direct, transformation and transduction are indirect. So plasmids encoded with drug resistance are naturally present in microbes before they've been exposed to an antibiotic. So transposons, or those jumping genes, also duplicate and insert genes for drug resistance into plasmids. So sharing of resistance genes accounts for the rapid proliferation of drug resistant species. So this slide is showing you some different mechanisms of drug resistance. So the first one is showing you an enzyme that is made, that are synthesized inactivating the drug. So it occurs when new genes are acquired. So this is an example of penicillin. If you remember, you have that beta lactam ring that is integral to penicillin working, or just being exactly what penicillin is. And then so these particular bacteria develop an enzyme called, or the genes for producing the protein, the enzyme penicillase, which is going to break one of those rings in that beta lactam ring. Then the next one shows the permeability, or the uptake of the drug into the bacterium, is decreased. So one of the ways that we have effective medications is we get it to where it needs to get to, and so, for example, inside the cell. So now the bacteria develops resistance to even getting that medication into its cell. You can also immediately get rid of the medication. So, in the third example, the medication actually binds to the receptors on the cell surface of the microbe, thus getting its effect to be done, getting that actual drug into the cell. But if you see, the bacteria can then encode for the process of actually forming a new pump that actually pumps out that drug that got in. Um, so that's a pretty clever way of evading medication. You can have the binding sites for drugs are decreased in number and the affinity. So this occurs via mutation or through the acquisition of new genes. So if you see here, one of the antibiotic classes actually works on the large subunit of the ribosome or the small, or both. And so in this case, it actually basically ruins or screws up the binding site on this particular medication. Is the large subunit of the bacterial ribosome, so that, hey, the ribosome can still bind and still read, and that bacteria gets the proteins it needs made. Um, Lastly, an affected metabolic pathway can be shut down. So if you remember back, there was, we didn't really talk much about it, but there are medications or antibiotics that will affect the folic acid pathway. So this is an example where the drug actually comes in and screws up that pathway, but the bacteria basically going the medication's mechanism of action. So, natural selection and drug resistance. The development of drug resistance and its long term therapeutic consequences. So, any large population of microbes will contain a few cells harboring drug resistance. So, as long as the drug is not present in the habitat, the genes are not expressed. If the population is exposed to drugs, the drug resistant population will have a selective advantage. So, offspring inherit the drug resistance, and then the replacement population evolves to have the drug resistant form as the dominant species. So to kind of piggyback on this point, if you remember, we talked about natural selection like at the very beginning of the class. And so, it's that giraffe that has the short neck isn't going to live. So that whatever's in that particular giraffe's genes that created that short neck, well, it's not going to be passed on because he's not going to survive to pass on his genes in the first place. So natural selection is, of course, only the strongest survive, or those with the necessary tools to survive. So in this case, for example, you have bacteria that naturally harbor the ability to resist some medications, but yet again, like the slide is saying, if you don't basically present that particular bacterial cell to an antibiotic, then it can't basically form drug resistance. This is one of the reasons we don't want to just give medications all willy nilly that aren't needed for the actual disease that you have. So if you see on this slide, the white cultures or colonies are they basically don't have resistance to the drug, but the red ones do. So after an hour of time, the particular medication actually kills the non-drug resistant cells, but not the drug resistant cells. So then, if you wait even longer, now the other cells are gone, and now they basically produced copies of themselves. So now you have a whole group of drug resistant cells to that particular antibiotic. So this is an urgent problem. So, the threat report issued by the CDC in 2013 outlines a potential catastrophic antibiotic resistant situation. We may enter a post antibiotic era where some infections will basically be untreatable. New and effective antibiotics have been slow to come to market. Antibiotics themselves are not economically lucrative, they're time consuming and expensive to develop. So basically, unfortunately, um, in a lot of ways in this world, we're very reactive instead of proactive. Even though we see the writings on the wall, we're probably not going to bother with really putting a lot of research into certain antibiotic treatments until it's, I don't want to say too late, but until it's a big problem. So, for example, we see with COVID right now, everybody's scrambling and putting all their efforts into finding a vaccine because really treatment for viruses aren't the best. It's best to prophylactically treat with a vaccine. So everybody's like all hammering to figure out, how to test, get the fastest testing kits out, and the vaccine. Um, well, that's what's going to happen probably with these infections. You're going to have more and more people who are susceptible to basically dying from a simple cellulitis infection, for example, and from some bug that we've been treating for years. And for some reason now it's not working. And the more we see it, and then when it becomes like a big crisis, because then that bug will be running around and invading and infecting people very easily. Now we're going to have an issue, and then we're going to bother to put our money and resources into it. So, urgent threats, we have C. diff, Clostridium difficile, carbapenem, which is an antibiotic, very strong one. So, carbapenem resistant Enterobacteriaceae. So, these are different, is a group of bacteria that affect the gut and cause diarrhea, really bad diarrhea. Drug resisting Neisseria gonorrhea. So, again, this is an effective agent or causative agent for gonorrhea. So, we have some serious threats. So that's like a little bit less severe than urgent. So multi-drug resistant Acinetobacter, drug resistant Campylobacter, fluconazole resistant Candida. So I talked about that Candida being treated with diflucan. So just yeast infections anywhere in the body. And concerning threats, so vancomycin resistant Staph aureus, erythromycin resistant group A strep, and clindamycin resistant group B strep. And those are really easy treatable bugs with these particular medications. So that would really suck that we can't treat them with them anymore. So new approaches to antibacterial therapy. So we're targeting iron scavenging capabilities of bacteria. We're also using RNA interface strategies. So these are small pieces of RNA that regulate the expression of genes. They're used to shut down the the metabolism of pathogenic microbes. So drug trials have begun to evaluate the effectiveness of synthetic RNAs in treating things like hepatitis C and respiratory syncytial virus, which is RSV. Some of you have probably had RSV before. It's very common for young kids to have it, and some older people. But there's I had a friend who's my age who had RSV not too long ago. So we have things that can mimic the defense of peptide molecules. So peptides have about 20 to 50 amino acids that are secreted as part of the mammalian innate immune system, called defensin, megainins, and proteagrins. Bacteria also produce defense peptides called bacteriocins and lantibiotics. So they insert into membranes and target other structures in cells, and they may be more effective than narrowly targeted drugs and less likely to foster resistance. So using bacteriophages, so Eastern European countries use mixtures of bacteriophages in medicine, but these drugs have never been approved for use in the West. So I used to work in clinical research, and so medications have to go through 4 trial phases before they can be released on the. Well, actually, they have to go through 3 before they're released on the market. And phase 4 is actually literally what effects are seen in the population while using that medication. Some medications then will get recalled in phase 4 because it's been out there for a while, and then we realize, hey, it's doing some stuff we didn't see during phase 1, 2, and 3. It has to go. It's pretty interesting if you're curious or ever want to work in clinical research. There's nursing jobs in that, but in basically the first phase is testing it out on, well, actually I forget each phase going from 1, 2, 3, but I definitely know that there's a phase where we tested on, I think it's phase 1, we tested on healthy adults who don't exhibit the disease that you're looking at, just to see what side effects that could come from the medication. Then you give it to people who do have the particular disease in question, to see what's going on. But it's a certain group of those people, and then from there, phase 3, I think we give it to a larger number of people, something like that. So, anyways, I was bringing all that up to say why some of these European countries use things that we don't. So, Biophage PA is used to treat ear infections caused by Pseudomonas aeruginosus biofilms. Other researchers are incorporating bacteriophages into wounds, and the advantage of bacteriophage is the narrow specificity. Only infect one species of bacterium. So remember, this isn't going to now affect our cells as well. It's just going to affect the one cells we're targeting. So, helping nature along, we have things like probiotics. So, these are preparations of living organisms that are fed to animals and humans to improve intestinal biota. So, this can replace microbes during antimicrobial therapy, augments biota that's already there. It's safe and effective. And is useful in the management of food allergies. So, these are probably things that you buy over the counter to help with your GI tract. I know several people who take probiotics. So, not to confuse probiotics with prebiotics. So, prebiotics are nutrients that encourage the growth of beneficial microbes. So, fructans encourage the growth of bifidobacterium in the large intestine and discourage the growth of pathogens themselves. And lastly, if you have me in A&P, we probably talked about fecal transplants. So, this is used to treat recurrent C. diff infections and ulcerative colitis. So basically, we're going to transfer feces from a healthy person to the person who is stricken with C. diff, and we're going to do this via colonoscopy. So the work is underway to develop a pill that contains the species to basically recolonize the colon so we don't have to go through the whole fecal transplant situation, which obviously can get a little messy.